Just to fill in while we're waiting for Alessio to come up, and Alessio is doing some really interesting work in developing inflammatory biomarkers and developing platforms in collaboration with the FDA and the EMA and the NIH, as well as other organizations, to see if they can develop platforms that are validated and reliable and could potentially be used as biomarkers for drug development and approval. So here he is. Alessio, can you hear us?
Yes, I can.
Okay, we're all ready for you. You've been introduced.
Thank you very much. And can I share my presentation?
Yes, you can.
Can you see my screen?
Yep, looks great.
Great. Well, thank you for the opportunity to present the work that we're doing. And I wish I was there in person. It didn't work for this year. But let me provide an overview about the FNH and some of the work that we're doing in neuropsychiatry as well. At the FNH, or Foundation for the National Institutes of Health, we really believe that science, especially these days, has the power of cure, but there is no single organization that can do this alone. So we were chartered by Congress in 1996 to support the mission of the NIH, and we built Bridge through Breakthrough, connecting leading researcher in working at the NIH, and not only, pharmaceutical companies and biotech, or any agency working in the life science space, and foundations, as well as patient organization. The work that we do go from goal to impact, meaning that we do listen and align. We are a convener. We are a safe harbor where people can share thoughts and ideas. We help designing the project. We engage with partners. We do the governance of the project from a scientific as well as financial point of view. And at the end of the day, we measure the impact as well. What people say about some of the partnership that we have done in the past and we continue to do is that they do see a lot of value in data, analytics, validation, infrastructure, a culture that we develop. The data, and I'm sure many of you, if not all, have used some of the data that we have generated, help to understand the biology in several aspects of neurodegeneration and beyond, and help understand the diversity from a patient perspective. They help with the analytics, meaning that we can develop new methods and approach that have been used by industry partners as well as by the greater scientific community, help to develop and validate novel targets and mechanisms. The validation is the next bullet point here. We have several nominated targets that help the drug discovery campaign from pharmaceutical companies, as well as biomarker and tools that have informed clinical trials. We did develop and we continue to develop infrastructure that can help bringing together all the data existing from a retrospective perspective and new. And certainly there's been a shift in the culture with the fact that now we can share ideas and thoughts and work all together. As an FNH, we work in all therapeutic areas, from cancer, cardiovascular, infectious disease to neuroscience. And today I'll mention some of the work that we're doing in neuroscience, as well as some of the work that we will do in neuroscience. And I will start with something that we actually did already. The FNH is considered by many a hidden giant. Most people know about the projects that are done by the FNH, but they're not familiar with the FNH per se. And one example is ADNI, the Alzheimer's Disease Neuroimaging Initiative, the largest and longest longitudinal study in Alzheimer's disease. Almost 20 years, the anniversary is going to be in October, of longitudinal data. And the FNH managed the public-private partnership that supported ADNI until the recent transition to the Alzheimer's Association that is managing the scientific board now. And ADNI data really transformed Alzheimer's research with almost 20 years of longitudinal data, working with over 150 partner organizations, thousands of publications, several thousands of MRI and PET scan data, and over 1 billion biosamples. So here the first thought that I have is, can we do something similar in the neuropsychiatric space? But let me go back to the neuroscience portfolio overall that we have. As you can see in this slide, we have multiple projects going on right now in Alzheimer's disease. One main project, and we're going to launch one soon in Parkinson's disease, one project in frontotemporal dementia, and one main project, the AMP schizophrenia, of course, working in schizophrenia. I was saying that we have a couple of large projects that we are going to launch in the next several weeks. One focused on Parkinson, one in ALS, and we are developing another project in Alzheimer's disease. And the point of this slide is we are doing so much in neurodegeneration, and we have just one project in neuropsychiatry, the AMP schizophrenia that I mentioned before. So what we are trying to do now is, can we do more in this space? There is so much that should be done in neuropsychiatry, and there are some ideas, there are some big challenges, and it has been harder to enter the space, but it's definitely our commitment and our intention. And as a matter of fact, and I'll tell you a little bit more about this project, we want to start with a small project focused on fluid synaptic marker for Alzheimer's disease and schizophrenia or major depressive disorders. Depression is another area where we are going to do something for sure, and I start talking with Andy about this project. As a matter of fact, as a thing that we want to do and develop, and I welcome your ideas and thoughts. Many other projects that are in the idea space. So this led me to the Biomarker consortium that I was asked to speak about today. The goal is to improving health through meaningful measurements, and the goal of the Biomarker consortium is to bring together industry leaders to validate and qualify biomarkers and other development tools to accelerate the development of these biomarkers and ultimately the approval to drugs that can hit the market. The Biomarker consortium bridge, as I mentioned, the gap that exists between basic research and clinical practice and the regulatory space. All the work that is done within the Biomarker consortium and within the FNH at large is pre-competitive, meaning that all results are released as soon as possible and everybody can access all these results as soon as possible. And all these projects are designed to have clear go-no-go funding gates. So if the project doesn't work, and we are actually in the process of ending one project as we speak, then we stop the project and we focus on something else. And right now we have about 40 projects that have been started since inception. And the Biomarker consortium has been quite successful over these years with 9 clinical tools being used in drug development, 5 FDA guidance documents, 1 qualified composite biomarker, many publications, citation. And I shouldn't be more clear. When we work with the NIH, we work with leading pharmaceutical companies, but we do work very closely with the FDA to take a seat around the table in any project that we do, because we want to have their buy-up as soon as possible in the process. So those are the companies that work with us within the Biomarker consortium. And I'll tell you a little bit more how you can engage with this group at large if you work in academia. Within the Biomarker consortium, as I mentioned before, the project that we have in execution right now focuses on neurodegeneration. We have one project in Alzheimer's disease, focused on plasma beta as predictor of a beta positivity in Alzheimer's disease. So this basically is the validation of the top performing blood plasma beta and tau assay, and the results are going to be announced in June 15, I believe. Neurofilament as a fluid biomarker for neurodegeneration. We are focusing on FTD to begin with. There's going to be a web announcement, I believe today, by the end of the day, since we received a letter of intent approval by the FDA. And then we are working on the pre-competitive analytical validation of SV2A PET in Alzheimer's disease to begin with. But as I mentioned before, we want to do much more in the neuropsychiatry space. And so the team decided to focus on fluid biomarker to begin with. Why fluid biomarker? Because we have a limited understanding of which synaptic biomarker or marker in combination are associated with disease pathology and clinical progression. We don't know which are the changes in synaptic markers that are a result of synaptic dysfunction, neurodegeneration, or mixed multiple mechanisms. Longitudinal assessment of these markers are lacking, and there is limited data on assay in blood and therefore limited understanding of the correlation between blood and CSF markers. So we're going to start with a focused context of use to develop prognostic biomarker clinical progression. And we're going to work in Alzheimer's disease to complement the ATN network, as well as in schizophrenia to provide new tools to understand the progression. And again, this project is in development. All this information here is developed as we speak. There is opportunity to change it in the next several months. What we want to do is understand the clinical utility and the biological context of different synaptic biomarkers in AD and schizophrenia. And the first aim is to assess our selected panel, both individually and collectively, is associated with clinical phenotypes in Alzheimer's disease as well as schizophrenia or other neuropsychiatric disorders. And we're going to leverage retrospective data as well. As a second aim, we want to investigate the impact of adverse conditions such as proteinopathies, neuroinflammation, mitochondrial dysfunction, and others. See how these comorbidities can affect synaptic dysfunction. And ultimately, I mentioned the SV2A project, and so we want to, in principle, create a relationship with that project and this project. So the last point that I want to make is, here's my email, and please, since I'm not in person, especially since I'm not in person, feel free to reach out if you have questions or comments. But the way the Biomarker Consortium work is these ideas come from multiple places. Sometimes come from industry, sometimes come from government, sometimes come from academic investigators. And so if you have an idea, if you have a project that you can develop, if you have already like a group of people working on something, and you need the buy-up of the FDA and working together with pharmaceutical companies, we are very happy to hear from you. So thank you again, and happy to take your questions.
So are there any key questions either in the room or online? We got one in the room. See if I can find the microphone.
Hi, Alessio. This is Lynn from Stalicla. You had questions from me prior, but I also have questions. A question for you. I see that one of the focuses for synaptic markers was, or the two focuses were neurodegeneration and schizophrenia. There's always this big debate around schizophrenia being classified as neuropsychiatric or neurodevelopmental, and now we know it's neurodevelopmental. But do you think that in biomarker consortiums we could try and integrate more of neurodevelopmental disorders, because we're seeing a lot of advancements in neuropsychiatry, in schizophrenia, bipolar disorders, and neurodegeneration, of course. And we sort of see neurodevelopmental disorders as more classically defined, because schizophrenia is among them, excluded from these efforts. And I think it really has its place in the space, both from a EEG standpoint, liquid biomarker, imagery.
Thanks, Celine. That's a great question. We worked in the past on a project on autism, the ABCCT, and I can send you some notes after. And when I started at the FNH, I was particularly passionate on autism, neurodevelopment in general. As we speak, we haven't really found the right group of people interested in autism or neurodevelopment in general, because the field is not ready. At least that's what we got out of our landscape analysis. But having said that, if you have something more specific in mind, some ideas of what a potential project should look like, feel free to reach out. I'm happy to discuss with you.
I think former Roche people like Chris Chatham and the former group working on neurodevelopmental disorders, they are very interested in integrating these efforts, and of course some industry players as well in the Fondation FondaMental. So maybe if we can push for neurodevelopmental disorders, that would be very nice.
That would be fantastic. Feel free to reach out.
Okay, so I'll pass over to my co-chair for the next question.
Yes, Alessio, can you tell us a little bit about the inflammatory biomarkers? I was hoping you were going to spend a lot of time talking about that. So do you want to share with us what you're doing in that space?
Yeah, thanks for your question. Actually, that project is the one that I referred before as project that we're closing right now, because the research team, as I mentioned before, everything is a team science approach. So it's not my decision or the FNA decision for anything that we do. And so the team decided the beginning of actually mid-April, so a month ago, to end the project and not to move in the next aims as a project per se. There is a lot of interest in inflammatory markers in neuropsychiatry, but that's not the right project. And so there is a strong intention to create something new. And so there is a lot of opportunity for people in this group to work together with us. But as we speak, we don't have a project yet. We're going to have in the next couple of months a pitch of ideas, and we're going to discuss what we are going to do moving forward. So there is a lot of opportunity to do something together in that space.
Okay.
Yes. Yes, there is a question, one question right here. Thank you.
Just try to understand it, since you said that we have, it's Carmine Pariante here, that we have possibility of getting into the space of this collaboration. If you could just maybe tell us exactly how to formally approach you for this, rather than just sending like a friendly email. Is there something, you know, how do we actually formalize such a collaboration with the people that are here and maybe other opinion leaders in the field of immunopsychiatry?
Yeah, actually, the first thing is just to send an email. We do a triage. I'm gonna direct you to the person working on my team on the Biomarker consortium, and she's gonna have like a first call. What do you pitch your idea? And she's gonna describe to you the opportunities that we have. But basically, the once we move forward, there's gonna be a pitch or a presentation to the steering committee that is gonna approve and decide which project they want to move forward with. Once that is approved by the steering committee, then there is the team that works on what we call the concept, that goes through some internal approval process through the Biomarker consortium leadership approval as well. And ultimately, we launch the project and we do the work that we were supposed to do. But again, the friendly email actually is the starting point as we speak of the process that we have.
Alessio, I don't want to push you too hard here, but why did you shut the program down?
I didn't shut the program down. It was a team decision.
Why was it shut down? You want me to ask it a different
Let's, we're getting a wee bit short of time. There is one question online. I think we need to make sure we get some of the folk online as well. And we're a little bit against the clock. So hopefully you can discuss some of those matters, Andy, afterwards, offline maybe. Alessio, could I fire in a question that's coming from Bruce Cuthbert, who's online, who thanks you for the great talk, and would ask, given the heterogeneity of schizophrenia, how do you identify specific subtypes for assessments in clinical trials? And that's a big question, probably with a huge answer, which we're all trying to answer. But if you could, a couple of sentences would be great.
Yeah, that's a big question. I agree. I mean, the schizophrenia project per se, we are going to have many data coming from that initiative. The first press release was done in October. There is the first set of data coming from that, but the majority of the data will come in the near future. Synaptic biomarker is not something that the team has been evaluated. That's why it was created as a standalone project. But I'm happy to follow up separately if I have your email. I don't see here, but I can send you some information about where we are right now with that specific project.
Thanks for the succinctness of your reply there, Alessio. And my apologies, time is running out. Many thanks for dialing in and giving us a chance to hear your thoughts, and also to hear that FNH is moving in some exciting directions, and we very much hope that everybody can work together to get some fantastic science done. Might I then move on to our next speaker? So we very much welcome Andy Lind, who I'm assuming is coming from Sweden. So Andy is a scientific liaison at Olinx Proteomics, who had an honourable mention this morning. Olinx has done much good work, particularly I think in the area of chronic pain research. But hopefully we're going to hear a little bit about how Andy thinks the omics world can help us in the area of neuropsychiatry.